Understanding Mpox. Mpox is a disease caused by the Mpox virus. The virus was first discovered in monkeys in 1958 and has since been detected in a variety of animal species. The first human case of Mpox infection was diagnosed in 1970 in the Republic of the Congo, located in Central Africa. Subsequently, Mpox has predominantly circulated in Central and West Africa, with transmission occurring between animals, primarily primates and rodents, as well as between animals and humans, and through human-to-human contact. In recent years, the rapid globalization, population movement, and deepening trade networks have contributed to the international dissemination of Mpox, resulting in outbreaks in various countries worldwide. In 2022, there was a large outbreak of Mpox. Mpox is a serious disease, but we can prevent it. We can also treat it if someone gets sick. Mpox spreads from animals to humans, which is called zoonotic transmission. Mpox can also spread from person to person through close contact. Symptoms usually begin within a week but can start 1 to 21 days after exposure. Symptoms typically last 2 to 4 weeks, but may last longer in someone with a weakened immune system. One of the first symptoms is fever. A few days later, a rash appears. Some people may have one or a few skin lesions, and others have hundreds or more. These can appear anywhere on the body. Identifying Mpox can be challenging because it can resemble other infections and conditions like chickenpox, measles, bacterial skin infections, scabies, herpes, syphilis, and medication-associated allergies. Co-infections with sexually transmitted diseases, such as syphilis or herpes, are also possible, making diagnosis more complex. Testing is crucial for timely care and preventing the spread of the illness. The preferred test is a PCR to detect viral DNA, ideally from skin, fluid, or crust from rashes. Swabs from the throat or anus can be used if no skin lesions are present. Blood tests are not recommended, and antibody tests are not useful for distinguishing between orthopoxviruses. HIV testing should be offered to adults with Mpox, and tests for other conditions like varicella-zoster virus, syphilis, and herpes should be considered where possible. Treatments and immunotherapy. Antibody-based therapeutics are showing promise in treating infectious diseases and are being actively explored for Mpox. Immune globulin, convalescent plasma, and neutralizing antibodies offer potential treatments for severe Mpox cases when antiviral drugs are insufficient. Individuals vaccinated with the smallpox vaccine produce neutralizing antibodies that may provide cross-protection against Mpox. Vaccinia immune globulin, which is against smallpox, has been approved in some countries and can be used prophylactically for immunodeficient patients who had contact with Mpox. Monoclonal antibodies targeting specific Mpox virus proteins have effectively neutralized orthopox viruses in animal studies, reducing viral loads and lung injury. Studies identified monoclonal antibodies targeting antigens that show broad neutralizing activity against Mpox. These monoclonal antibodies provided strong protection against lethal doses of Mpox in mice, even in immune-deficient models, showing greater efficacy than immunoglobulins or convalescent plasma. Therefore, MAbs are likely to be more effective in future clinical treatments for Mpox. Currently, antiviral options for Mpox are limited, with cidofovir and its derivative brincidofovir being some of the most notable treatments. Tecovirimat, approved in the wake of the 2022 outbreak, targets the viral protein VP37 and shows promise in reducing viral spread. Other treatments under research include nucleoside analogs like ribavirin and novel compounds that disrupt viral replication or entry into cells. These medications work by stopping the virus from replicating in the body. This can help shorten the course of the illness and reduce the severity of symptoms. Scientists are constantly researching new treatments for Mpox. The goal is to develop safe and effective treatments that can prevent serious illness and death from the virus. One of the most effective ways to prevent Mpox is through vaccination. The Jynneos vaccine is safe and effective against Mpox. It is given in 2 doses, 4 weeks apart. Individuals who recovered from Mpox do not need any Mpox vaccine doses. Vaccination is especially important for people at higher risk of exposure to Mpox. This includes healthcare workers, laboratory workers, and people who have close contact with infected individuals. In addition to vaccination, there are other important preventive measures. Avoiding contact with infected animals is crucial. Rodents including squirrels, rats, mice, and prairie dogs, and primates including monkeys are known to transmit the Mpox virus. This includes avoiding sick or dead animals in areas where Mpox is present. It is also important to practice good hygiene. Washing hands regularly with soap and water is essential, especially after contact with animals or potentially infected materials. Using alcohol-based hand sanitizer is a good alternative when soap and water are not available. Scientists are continually learning more about Mpox. Ongoing research is vital to improve our understanding of the virus and develop better ways to prevent and treat it. One area of research is the development of new vaccines and treatments. Another challenge is the potential for the virus to mutate. Viruses can change over time, and these changes can sometimes make them more difficult to treat or prevent. Scientists are closely monitoring the Mpox virus for any signs of mutation. Global collaboration is essential to control Mpox. Sharing information and resources between countries is crucial to track the spread of the virus and implement effective control measures. Public health education is also vital. By understanding the risks of Mpox, how it spreads, and how to prevent it, we can all do our part to protect ourselves and our communities.